My name is Muhammad Dini Irfan bin Muhammad Nur Hisham. My metric number is E23A2608.
Hello, my name is Muhammad Nafiq Syami bin Abu Sairi. My number matric is E23A3091.
My name is Nur Shawani binti Muhammad Rajab. My metric number is E23A2870.
Hello, I am Nur Anati Husna binti Isha. My number matric E23A2692.
My name is Mazatu Sofia binti Sabirin. My metric number is E23A2644.
Hi, my name is Muhammad Hakimi bin Kamarul Zaman. My metric number E23A2638.
Hi, my name is Narka Shirah binti Jamdain. My metric number is E23A2728.
My name is Atirah binti Muhammad Fuad. My number matric is E23A2774.
My name is Muhammad Azri bin Zaharul Nazli. My metric number is E23A2941.
So, today we are gonna talk about the Nobel Prize winner, Sidney Brenner. But who is actually Sidney Brenner? He was a biologist from South Africa where he was born in the town of Germiston in South Africa on 13 January 1927. He was educated at University of the Witwatersrand, Johannesburg, South Africa. Among the awards won by him are Nobel Prize in Physiology or Medicine, Copley Medal and more. He died in 5 April 2019 at Singapore.
The History of Winning Experiment by Sidney Brenner. He was awarded the Nobel Prize in Physiology or Medicine in 2002. He introduced Canohabditis elegans or C. elegans as a model organism. He chose C. elegans as an ideal subject for studying development and neurobiology at the cellular level because of its simplicity, well-defined anatomy, and its short life cycle. He managed to develop genetic mapping technique for induced mutation, map the genetic structure, and laying the groundwork for understanding how genes control the development of C. elegans.
The contribution to developmental biology. His most significant contribution was cell lineage analysis of C. elegans. He observed the fate of every cell from embryo to adult. He managed to demonstrate the deterministic nature of cell division and differentiation in the organism. His other contribution was apoptosis or programmed cell death. Apoptosis is important for development and maintaining cellular homeostasis. His foundational research for identifying key genes and regulating apoptosis, then continued by Horvitz and Salston, has profound implications for various fields.
We proceed to his genetic and molecular technique in the experiment. He introduced forward genetic techniques. This technique required the use of mutagenesis skin in C. elegans to identify mutation that causing biological defects. This technique revealed that gene is important for development. He also led this team to perform detailed study on the nervous system by taking advantage of C. elegans transparency. His map all 302 neurons in hermaphrodite worm and provide critical neurobiological resource.
Obviously, his research has its impact and its own legacy. He revolutionized biological research by demonstrating to uncover fundamental biological principles using simple organism model. From genetic and neurobiology perspective, he provided insight into human biology disease. Use of C. elegans has now been standardized in laboratories worldwide.
Now, let's dive into the diverse application of Canohabditis elegans as a disease modeling. First, as a neurodegenerative disorder. C. elegans recapitulate key features of human pathologies, including protein aggregation, neuronal dysfunction, and progressive neurodegeneration. These models have helped identify genetic modifiers, toxic protein types, and potential therapeutic compounds for diseases such as Alzheimer's, Parkinson, and Huntington's. Second, as a cancer and metabolic disorder. By manipulating oxygenase, tumor suppressors, and metabolic regulators, researchers can induce tumorigenesis, metabolic dysregulation, and other disease phenotypes in C. elegans. This model enables the study of cancer progression, metabolic pathways, and therapeutic intervention, and provide valuable information about human cancer biology and metabolic disease. Next, health and drug discovery, which is in the effective screening. C. C. elegans-based assays enable rapid screening of large compounds to identify molecules with pharmacological effects on various biological processes or disease phenotypes. Automated platform and image analysis techniques improve the efficiency and throughput of C. elegans drug screening, facilitating the discovery of new drug candidates and chemical probes. Last but not least, help in medical studies. The elegance model provide a platform to study mechanism of action of drug and elucidate their effects on molecular pathways, cellular processes, and organismal physiology. Through gene manipulation and pathway analysis, researchers can refine drug addiction, identify downstream target, and uncover potential side effects.
The Advantage of Chenohepatitis elegans by Sidney Brenner. In the world of medicine, Brenner's groundbreaking research has revealed new treatment targets. Potential targets for the creation of innovative medication have been found through the discovery of particular genes and pathways. Modulating apoptotic pathways is one such instance that show potential for treating illnesses marked by dysregulated cell death or survival. Besides that, C. elegans is an excellent model for aging research due to its ease of maintenance, transparent body for observation, genetic similarity to human, high fertility rates, and availability of molecular biology tools. Its short lifespans and small size make it favorable for screening anti-aging drugs with reduced experimental costs.
I will explain the disadvantages of Sidney Brenner experiment. The first one is simple organism. The number of cells in C. elegans and human is different. Humans have more cells than adult-sized C. elegans. It is estimated that human cells have 37.2 trillion cells, while adult C. elegans only has 1,000 cells. This causes the selection of C. elegans as a model in this experiment to encounter some difficulties due to the fact that this organism is too simple compared to humans. The second one is procedure weaknesses. The procedure used by Sidney Brenner is a little weak because the procedure used cannot detect mutations. Mutation refer to a genetic change that result in variation in the physical characteristic of C. elegans. This mutation affect slow growth. This is because the isolation process used is not very sensitive to detect such mutation.
The Chenohepatitis elegans genome, which contains two-thirds of all human disease genes, has been used as a model system for studies on aging, age-related diseases, longevity mechanism, and drug screening. This review reviews C. elegans mutants exhibiting increased lifespan and age-dependent change under dietary restriction, providing evidence for its use in research on aging prevention. C. elegans are valuable models for research in aging due to their age-dependent physiological change. C. elegans shows a decline in their anatomical and functional features, including tissue, integrity, motility, learning, memory, and immunity. Age-dependent change in C. elegans includes diminishing nuclei integrity and increased nucleoli size, varying depending on tissue types. Mitochondria undergo structural and functional change, including fusion and fragmentation, affecting DNA copy numbers and oxygen consumption rate. The unfolded protein response of endoplasmic reticulum capacity is reduced in C. elegans, leading to increased misfolded protein in age-related diseases. Age-associated molecular change provide valuable biomarkers for aging, with change in gene expressions affecting lifespan but decreasing with age. Studies have identified over 50 genes controlling aging in C. elegans with many homologs in other organisms. This study discusses longevity mechanism related to transcriptional regulator of metabolic networks and epigenetic regulation in C. elegans.
Human discusses a significant case study involving bacteriophage T2 infection of Escherichia coli cell to explore the mechanism of protein synthesis and the role of messenger RNA, mRNA. The study primarily aimed to understand how genetic information is transferred from DNA to ribosome in the context of fish infection and how this process differs from normal cellular conditions. The experimental design, the researcher used isotopic labeling to track the synthesis of RNA and protein post-fish infection. They grew E. coli in a medium containing heavy isotope and then transferred the cell to a medium with light isotope immediately after infection with the T2 fish. This allowed them to differentiate between pre-existing and newly synthesized molecules using density gradient centrifugation. mRNA characteristics. The mRNA in infected cells was shown to have a rapid turnover and a specific nucleotide composition that matched the fish DNA. This RNA fraction was physically distinct and had a size large enough to code for long polypeptide chain. Contribution experiment to medicine. Gene expression insight. Understanding mRNA role paved the way for advancement in gene therapy and the development of our mRNA vaccine as it provided foundational knowledge on how genetic information is expressed in cells. Biotechnological application. The insight from this study contributed to the development of recombinant DNA technology and genetic engineering, which are crucial in producing pharmaceutical and therapeutic protein.